In this video, we'll look at experimental design, in particular some biases. What we'll cover in this video is the placebo effect and some causes of sample bias. And then keep in mind that for the second exam, there's a website to go and look at that discusses a variety of misuses of statistics that will be tested on the exam. So the first thing we're going to do is talk about the placebo effect, which you've probably heard of to some extent, but we'll look at it in more detail. So the placebo effect is this idea that receiving some kind of treatment causes improvement even if the treatment itself is a sham or a fake. So for this reason, to test the efficacy, that is effectiveness, new treatments must be compared not to untreated controls, but to placebo-treated controls, right? The appropriate control is not someone who's getting nothing, it's someone who thinks they're getting the same thing as the drug, but are in fact getting nothing, or nothing chemically active. But if the recipients, or the people getting the drug, know whether they're getting the real drug or the placebo, then this whole process fails because they would be able to identify what's going on. So for this reason, the individuals getting the drug must be blind to which treatment they get. The people getting the treatment can't know. And in fact, even more than this, there's an extra kind of blindness we need to add, and this all has to do with a particular horse called Clever Hans, who could do math. So here's the biography of Clever Hans. So he's from the early part of the 1900s. Hans the horse is owned by a guy called Van Osten, who's this guy here. And Hans was a horse that could do math problems. So for example, he would go on tour and someone would ask the horse, what is 2 plus 2? And then the horse would tap his hoof on the ground. And then it would stop when he got to 4 and he'd done the correct answer. So an investigation in 1904 found that there was no fraud or tricks, right? Van Osten wasn't like giving signals to the horse or something like that to tell the horse when to stop. So a little later, a guy called Funkst followed up with an investigation of his own, and he did more detailed kind of treatments. So he found out that the horse, Hans, gets the answers correct even if Van Osten is not involved, right? So if the owner kind of goes around the corner and is not visible, is not actually present, the horse can still get these math problems correct. However, if Van Osten is present, it gets a little bit more interesting. So the horse, when Van Osten is present, usually gets it right about 89% of the time. But if Van Osten is there, but he has on headphones or something like that, so he doesn't know the question, then the horse's ability drops down to 6%. So that's kind of strange, right? The horse is able to get it right when Van Osten is not there, and when Van Osten is there, but doesn't get it right if Van Osten is there and doesn't know the question. So it turns out what was going on is that the horse needs to see someone who knows the question and the answer, and he's watching to see when they subconsciously relax, right? So everybody's kind of tense as the horse is tapping out the numbers. Once the horse gets to the correct answer, then people subconsciously relax. And if Van Osten is there, the horse is looking at him. If Van Osten is not there, the horse is looking at everybody else, and they're doing the same thing. And of course, if Van Osten is there and he doesn't know the question, then he doesn't know when to relax, so then the horse gets it wrong. So having figured this out, Funkst is then able to train himself to have 90% accuracy when he acts like Hans, right? So he can go in front of a crowd of people, put on headphones, a math question is asked, he taps out the answer, he looks for when people relax, and he's able to get 90% accuracy even if he never knows the question. So he's able to simulate the horse. But a follow-up to this is he's actually then unable to suppress the cues that he just figured out existed even after he knows they're present. So when he goes back and works with the horse and deliberately tries to suppress his relaxation, it doesn't work. The horse is still able to read him and is still able to get the right answer even when Funkst is deliberately trying not to send the signal to the horse. So of course, having figured this out, this doesn't actually stop the horse's career, because once we figure out what's going on, that often has no effect on what people are willing to watch for entertainment. But this is important for our placebo effect because the inability to suppress cues and information that we have when we're dealing with other people is very important. Because of this danger of these subconscious cues that we can't control, in any experiment, the provider of the treatment must also be blind, right? The person giving the drug cannot know whether it's a placebo or the actual drug. Otherwise, that means the patient might know if it's the placebo or the actual drug, no matter how hard the person giving it to them tries to prevent that. So for this reason, both the person getting the drug has to be blind, and the person providing the drug has to be blind, and that creates something called a double-blind experiment. And essentially, in order to have an experiment that successfully eliminates the possibility of the placebo effect, it must be double-blind. A double-blind experiment isn't always possible. So for example, back in the day when I used to rent my body to science and test out experimental medications, one of the things I took was a drug called, or a dye, called methylene blue. They had noticed that perhaps it helped people's moods, so I got paid to take this drug and fill out a questionnaire. And then they also, some days, gave me a placebo instead. But for that experiment, it was pretty obvious to me which was the real treatment, which was the placebo, because this is a very, very powerful dye that had a dramatic effect on my physiology, in a sense. So that was a particular study where the placebo wasn't really possible, even though, of course, it would have been ideal, because I was filling out questionnaires about my mood at a certain time each day after taking either this or the placebo. But in general, you cannot trust a medical study unless it's double-blind. A follow-up to this would have been designed to be double-blind. Now, that doesn't mean placebos aren't important. Placebos are actually prescribed all the time, right? Doctors do this. They prescribe vitamins for annoying patients, right? Vitamins, if you have a healthy diet, are almost totally useless unless you're pregnant. And doctors also prescribe vitamins for viral infections, and that's not really something that does any good. Doctors also prescribe antibiotics for viral infections, and those only work against bacteria. And the placebo effect is real, right? The entire alternative healthcare industry, all those supplements and herbal things, there's essentially almost no evidence that any of them work, but people who take them think they work, and thereby they actually sometimes do work by the placebo effect. The same thing goes for chiropractic. Chiropractors treat people for back problems, and that seems to be kind of legitimately effective. But chiropractors also sometimes treat people for things like liver problems or acne problems, something for which adjusting their back makes no physiological sense, but because they've been treated by a trained professional who is full of confidence, often those troubles actually improve on their own, and that's going to be because of some sort of placebo effect. Okay, moving on to thinking about various biases in science. Let's look at a couple of related ones, the cherry-picking problem and the file drawer problem. So the cherry-picking problem is this idea that if you do a series of studies, you're only going to report the results of the studies that are favorable to you. So for example, a pharmaceutical company does 2 studies with some sort of drug. Study 1, p-value less than 0.05, the drug works. Study 2, p-value larger than 0.05, the drug does not work. So then what do? They tell the world about the first study, and then they just kind of forget the second study was ever done and don't tell anyone about it. And what that results in is if you look in the published literature of studies on this drug, all of them say that it works, right? Because 100% of the published studies have this p-value of less than 0.05. The public doesn't even know about this. So a solution to this is to create a database of all the trials that are started and only allow the publication of trials that are in the database. And then what researchers can do is they can follow up with these drug companies. They can look at all the studies that were started, that were registered in the database, and then look to see if any have been left out, right? If this pharmaceutical company registered this and then never published it, we can go and ask them, well, what are the results of this study? So this system is actually in place for clinical trials, because in 2004, the New England Journal of Medicine, the Lancet, Annals of Internal Medicine, and the Journal of the American Medical Association agreed to use this public database of drug research trials. Only studies that were registered before they even started are allowed to be published in those journals, and those are high-level journals that pharmaceutical companies and medical researchers want to publish in. So there's an incentive to do this. Now, this is not a solution for all the other parts of science, right? This exists for medical studies because there's a lot of money involved, but it does not exist for other things like psychology, ecology, other fields where if someone has an agenda, they could do multiple studies and only publish the ones that support their favorite hypothesis and just bury the ones that don't. A related problem is called the file drawer problem. So it's going to have the same effect on science, but the motivation is going to be more benign and less nefarious. So in the file drawer problem, we're only going to report or publish papers that have significant results. So for example, say a scientist does 2 studies on sexual selection in flies. That's one of the types of research I do. In the first study, turns out there's no sexual selection, right? P larger than 0.05. In the second study, there is sexual selection, P less than 0.05. And so the researcher has 2 studies that they've completed, and then they kind of put all their effort into this one. And this is not kind of cherry picking with some sort of intent. It's a file drawer problem, right? So everybody is limited in time. Nobody has enough time to publish all the studies they do. This is the one that has a higher chance of getting published and noticed. This one, it's going to be hard to get a journal to accept a non-significant result. So you're going to put this one on the front burner and publish this because it's going to get published. And you'll put this on the back burner and you'll get around to it someday. But of course, someday never happens. But this actually has the same result, right? 100% of the published studies will actually say that sexual selection is present in flies. Even though this study and this study were both performed, if only this one is published, all of the publications say that sexual selection is occurring, even though it only seems to happen half the time. So the solution to this is to make publishing negative results acceptable, right? To make it kind of more socially acceptable to publish papers where you did an experiment and didn't see the thing you were looking for. Now that actually hasn't happened yet. This DA selection example I have here because back in the day, I did an experiment with an undergraduate researcher. We looked at directional asymmetry. So that's basically with flies. We were looking at their wings to make their left wings bigger than their right wings. And she did 15 generations of artificial selection, which always works when you select to change an organism. But in her experiment, it did not work. We were not able to make them more asymmetric. So that's an interesting result because asymmetry evolves in nature, right? Narwhals are asymmetric. That horn they have is their left incisor. But when done in the lab, asymmetry seems to be the only thing, essentially, that cannot be selected for. However, getting no response is P larger than 0.05, a non-significant result, even though it's an interesting result, turned out to be harder to publish. So we did end up publishing this because this coincided with when I first got hired at Long Beach. So of course I needed publications to get tenure. Also, that student was starting her graduate career in Arizona, so it was kind of nice for her to get a publication to get things going. But now, later in my career, would I be as motivated to put all that work into a study like this? Or would I just kind of put it on the back burner and focus on my more recent studies, which have actually shown things like this? I'm not like a morally bad person, so I'm not going to do cherry picking, but I don't have time for everything, so I might end up doing this. And, take my attitude and my lifestyle and copy it several thousand times, and you end up with something that might actually be creating a real problematic bias in science. Another bias is a bias in observed effect sizes. The chances of getting a significant difference depend on the size of the effect, right? If you have a difference like this versus a difference like this, you'll be able to detect this difference much better than detecting this difference. So when you do a bunch of experiments, you're not going to see the small differences, but you will detect and see the large differences. So essentially we see bigger effects and bigger differences better than smaller ones. This creates something called the Beavis effect. So we think the real effects that we see, the ones we can get statistically significant results for, are all large when in fact many of them are small. And this is a form of bias in that it's causing us to misunderstand what the typical magnitude of the effects out in nature really are. This is actually a big problem for genetics studies, particularly ones using microarrays and QTLs. These are studies like this. This is a microarray, where they look at a large number of genes looking for some sort of trait, like what are the genes for breast cancer risk, or what are the genes involved in heat shock response. Now, when they do that, and they're looking at differences in expression patterns in two different treatments, they'll be able to see the large differences, but not see the smaller differences. And what that means is they'll identify some genes, but miss some other ones. And the ones that are identified are the ones that have large effects. And so what that means is we actually have no idea how much of the true risk is in small genes that are missed. We think there's a smaller number of genes with larger effects. So we end up completely misunderstanding how genetics works. We think that genetics works because there's a small number of genes that each have a large effect, but in reality, there's a small number of genes with large effects, and then potentially many, many more genes that have smaller effects. And maybe that second set of genes adds up to a much larger overall effect than the first set. And unfortunately, there is no good solution for this problem. Let's quickly look at a few more biases in biology and ecology. So one kind of conceptual bias is shifting baselines. So if we think about how many sea turtles are in the Gulf of Mexico, the number in 2009 versus the number in 2010, that number went way down, right? The Deepwater Horizon spill killed a bunch of them. But even then, we're thinking 2009 is the normal number, but back in 1710, when people first kind of from Europe arrived in the Gulf, there are ship's logs of the captains at the time that talk about the waters being so full of sea turtles that you could hop off the boat and walk to shore on the backs of all those sea turtles. They're just so common. They're like vermin. Well, that is not at all how we think of sea turtles now, right? But it's perhaps how they should have been, and it's how they were. But because things change, and we just get used to things, we don't even realize that things have changed and are different. Another bias that applies to lots of people, in particular people in places like Los Angeles, summarized by this book called Last Child in the Woods. Most of us today have very, very little direct, unstructured contact with nature, right? A time to see how animals and plants work without someone telling us exactly what to do. I have this movie here because there's a really interesting conversation at a dinner table in a family where they're talking about evolution and they're talking about the livestock that they have. And in a family that has livestock, evolutionary ideas are very obvious. But if you've lived your whole life and you've never been on a farm and you've never looked at a population of organisms, it can be very easy to think that evolutionary ideas are not true, or evolutionary processes don't exist in the real world, and thereby end up believing things like creationism or stasis or things like that, which are actually not true. And it's because of the kind of limited exposure that urban dwellers now have to nature, which makes it particularly hard for us to understand nature if we just don't have the experience with it. Some more biases that have less to do with humans and more to do with animals involve the fact that some animals are easy to study, some are more difficult, and actually our technology and our ability to see what they're doing can create biases. So for example, for a long time we thought that birds were monogamous, right? A male bird and a female bird get together, they lay a bunch of eggs, they have a nest, and that's one big happy family or something. But then later, when we got DNA technology, and we were able to make observations that we previously couldn't make, we were able to see that, well, actually a lot of the chicks coming out of those eggs had a different father from the male that was helping to raise them. So we actually had a bias in what we could see until we developed new technology, and then all of a sudden we were able to see a type of behavior that had been previously unknown. Another bias that complicates some of these ecology studies is the use of bait traps. So a lot of times we set out traps overnight, put a little food in there, catch the animal, take a blood sample, take some measurements, whatever. So the animals get a food reward, and it turns out they often like it, which means the next night they may be more likely to come back and get sampled. So most of the sampling that we would be doing is going to assume sampling with replacement, right? The probabilities are not changing as we're taking our individual samples out of the population. But if individuals who have already been caught are more likely to get caught again, that messes up that whole thing and can actually cause a bias, which can mess with our ability to estimate the population. Now, one thing to note about all of these things is that bias is different from noise. Statistics can handle noise, right? Noise is unusual events. Noise is natural variation. Noise is measurement error. And statistics can handle all of that, right? That goes into the variance, that goes into the standard deviation, goes into the standard error. Bias is different. Bias is undetected. Bias is not in our equations. And so the mathematics of statistics cannot handle bias. Well, unless we kind of have recognized it by knowing about the causes of bias, we can actually just not even see it at all. We'll do our statistics, we'll get some sort of result, and it may be completely erroneous, not because of randomness and weird things, right? Statistics can handle that, but because of consistent, unrecognized biases, statistics does not do a good job of handling that. Continuing with biases, there's a bias in polling data if we're thinking about studying human opinions. So magazine polls, who even reads magazines anymore? The internet, that's fine, but only some people use the internet and skews younger. Right, most telephone polls are by landlines only, so people like my father will answer that, but I won't. I don't have a landline and haven't had one for 20 years. And a particularly interesting type of bias in polling data is something called the Bradley effect. So this goes back to a governor election in the state of California. There was a candidate called Bradley, and he was an African American. And in the polling data leading up to the election, he was going to win. But then when the election happened, he turned out not to win because a lot of people who had said they were going to vote for him, when they got in the privacy of the voting booth, they basically exercised their kind of anti-African American tendencies and voted for the other candidate. This bias was basically because people often tell pollsters what the pollsters want to hear, or tell them what makes the person being polled look good in the eyes of the pollster, right? People with unpopular opinions, like being a racist, are not interested in expressing that when they're polled, and that can contribute to something called the Bradley effect. We may have actually had a similar version of this lately, right? So in the most recent presidential election, when Trump won, the polling leading up to that is that he was going to lose, and that may have been because a lot of people called on the phone and they're asked to choose between Hillary Clinton or Donald Trump. They would have said Clinton, or they would have said undecided, when in fact they were leaning towards Trump. I actually have essentially a close relative who was a huge Trump supporter the whole time leading up to the election, but she didn't tell anyone because she knew that he was an unpopular candidate. But she certainly voted for him when the time came, even though she would have never told someone asking her who she was going to vote for honestly. There's also bias in the census if we're thinking about another way of assessing humans. The Constitution mandates that the U.S. count every person in the country every 10 years. And so we want that population data in order to apportion congressional districts and have highway funding and things like that. But we also know we're going to get a biased sample, right? There are certain populations of individuals who are maybe less likely to be home when the census taker comes by, or are less likely to open the door and sit down and have a fully honest conversation with someone who says they're from the government. So if we know that we're undercounting certain groups of people, should we adjust the samples to reflect this? And if so, how do we adjust the samples to reflect this? And this actually leads to one of the differences between the political parties right now. Democrats are more in favor of a thing called statistical sampling, which is recognizing this deficit, this bias, and adjusting the values to try to fix them, versus Republicans who think that maybe that leads to manipulation of the data, and we should perhaps just do our best to count everybody and not try to do any of these manipulations. So the bottom line is, how do we adjust the values to increase the number of minorities and poor from the ones we measured, because those are the groups that tend to be undercounted. And if you're a Democrat, you're probably going to be more aggressive about that adjustment. If you're a Republican, you're going to think that opens the door to too much manipulation. And then that leads us to actually have this debate every 10 years about how to actually get the best representation of the population from the sample that we're taking that we know is biased. And we don't have a good solution to that yet. We just keep on trying to figure it out every 10 years. The last bias that I want to mention is this confirmation bias, which was in the first part of the course. Basically, people are better at remembering things that match their previous beliefs than things that don't match, right? See, and actually this cartoon is a little inaccurate, right? This isn't even a conscious thing. This cartoon is making it seem like confirmation bias is a deliberate attempt, right, to seek out facts to support our opinions. It's more insidious than that. It's the fact that we actually remember facts that support our opinions better than facts that counter them, not even consciously. And in fact, this is a danger of social media, which is how most of us get our news these days. Social media is essentially going to show you what you want to see because they want their customers to be happy. And when people are happy, that's when they're seeing things that make them feel like they're correct about the world, right? Things they agree with, not things that challenge their assumptions or their beliefs. And in fact, there was a study, this is a few years ago now, Facebook actually did an experiment on a bunch of people. What they did was they manipulated the news feeds of about 700,000 people, and they gave them either good news or bad news, or more good news or more bad news than usual. And then they looked at what people posted afterwards, right? And it turns out that when people were given good news or things they agreed with, then they posted happier things, and they seemed to be happier customers. When they were given bad news or given things that disagreed with what they already agreed with, they then posted less happy things. So of course, what does Facebook want? Facebook wants people to enjoy their product and be happy, and because they can control the feed, this is the danger of social media, right? They show you what you want to see, but in fact, even more than that, they're going to show you what they want you to see, right? If they want you to be happy, they'll show you happy things. If they want you to be suspicious, they'll show you things that make you suspicious. And this is a danger now in the real world. It's a bias that we're now all prone to when we get our news from just one source.